everyone. If you're a returning subscriber, welcome back. If you are new, welcome to my channel. My name is Dr. Shingo Mufawa. Guys, if you haven't subscribed, please do so. We want to create a community here. In today's video, we are going to discuss the full protein synthesis process in a simple step-by-step format. Let's go from DNA to protein. So, what is protein synthesis? Protein synthesis is how your cells build up proteins like enzymes and hormones using the genetic instructions from the DNA molecule. Protein synthesis happens in two stages, which are transcription and translation. Transcription, by definition, is the process by which a messenger RNA molecule is synthesized using the DNA molecule. Transcription takes place in the nucleus. My advice is, when you write this process, do so in point form. Transcription starts with the DNA molecule unwinding, and the double-stranded DNA unzips, weak hydrogen bonds breaking to form two separate strands. One strand is used as a template to form the messenger RNA using free RNA nucleotides from the nucleoplasm. The messenger RNA formed is complementary to the DNA molecule. Messenger RNA now has the coded message for protein synthesis. The messenger RNA synthesized moves from the nucleus to the cytoplasm, where it attaches to the ribosome. Please note that during transcription, only one strand is used as a template. And when talking about the use of free-floating nucleotides, you are expected to mention that they are RNA nucleotides and not messenger RNA nucleotides. Also note that there are no base pairings here because the messenger RNA molecule is single-stranded. Now, let's talk about translation. Translation is the process by which different amino acids are assembled to form a specific protein. The process of translation takes place in the cytoplasm on a ribosome. Sometimes the examiner will ask you for the organelle where the process of translation takes place, and this organelle is the ribosome. Now, the process of translation takes place in the following way. Each transfer RNA molecule carries a specific amino acid, and when the anticodon on the transfer RNA molecule matches with the codon on the messenger RNA, the transfer RNA brings the required amino acid to the ribosome. The amino acids become attached to each other by peptide bonds to form the required proteins. Be reminded that the amino acids are attached to each other through peptide bonds and not polypeptide bonds. Students usually confuse these two. Also, note that the examiner does not expect you to memorize the names of the specific codons, anticodons, and their amino acids. That's the full journey from DNA to protein. If you learned something new, give this video a thumbs up. Don't forget to comment, share with your study buddies, and subscribe for more tutorials. See you all in the next video. Bye.